Your camera's off. So
Camera's off?
says.
Yeah.
Oh, yeah.
Just click the camera again.
Yeah.
Click the.
Yeah,
I think I clicked it twice, sorry.
Okay, well, pretty much these are the methods that we developed until now. And I don't know now, maybe it's not moving.
Click back on.
Okay.
You good?
Okay, yeah, all of this is published if you want to go and find any information about that, except for helicase dependent that is still in grounds. So coming back with some details, there was too many inconsistencies in the early detection method PCR based because the difficulties of the tissue. And at that time, working with Binoy and Matthews Paret, we came out with the direct trapping of the rhodosite virus into the PCR tube. Is working lovely. I understand that many labs adopted now. I think Kevin also is doing it, right? I don't know, you modify it and then, yeah. So everyone has been doing a tweak according to the lab, but it's working lovely. And the principle is that you can absorb the capsid protein of the virus into the PCR tube because the difference in charges. Then we went, trying to move to the field to assist the breeders. We developed this LAMP method. Is a method that is quite nice. You don't need the thermocycler, but it's semi-field deployable because you still need some equipment. And in here, at this point, we realized that we were working in the RNA-3, something that you guys pointed yesterday, that the RNA-3 is a good spot. So for diagnostics, and yesterday I said good because we have been working the RNA-3. Initially, we moved to RNA-4, but then we realized that the RNA-3 actually was much better. After that, we noticed that there were too many primers that people developed because this inconsistency. So there were more than 17, and then we say, okay, we have a technology from 2013, and then we say, let's build an artificial positive control that works for all these primers and probes. And this is a work made by Rafaela, and worked very nicely. At that point, Kevin complained to me, and he said, no, I don't want plasmid in my lab. It's going to create too much contamination. I don't want plasmid flying around. And then I remember I asked Kevin, what do you want me doing? And they said, I want them. Someone has a microphone open. And then Kevin said, I want it in the matrix, something solid. Here it is, Kevin, so now you can use it. So you can put it in this little piece of paper, which is 1 millimeter diameter. And it's fixed there, it's not flying, it's not going anywhere, and it worked perfectly. So you can do LAMP, you can use probes, you can use the PCR for the insect, everything is working well. And if you want the sequence and doing your own plasmid, is in the NCBI. So the only thing that you need to do is taking that sequence, ordering your plasmid, and it's working very nice, very consistent. All of this information and working with Drushalek, then we came out with the life cycle of the rhodosite. This diagnostic play a very important role in defining this. Is something that you know already very well. And then we noticed that there was a problem. The first 4 RNAs were reported by Langley, and then develop came with the other. And then that's created a problem if you wanted to, well, not a problem, but some inconsistencies in some of the molecular biology tools. So I decided to sequence the Oklahoma isolate, which is quite aggressive, I would say. And the sequences are there in the gene bank. This was a piece of collaboration with my friend Pierre Koenig from the Netherlands. And then in Europe, you need to know they are very concerned about having road rosette. So there's a lot of collaboration going on from my lab with some colleagues in FERA, UK, the Netherlands, Italy. And something that you guys were talking yesterday as well, it was about the movement of the road rosette. Well, we found that in 1984, Drurik presented some data in his master thesis, and he pointed out that there was tropism in this virus, and then the virus was moving toward the roots. At that time, working with Binoy, it was winter time. He said, let's go to the field, and it was January, it was very cold. He said, let's get some roots from the dormants. And they test positive. So we say, okay, let's design this experiment. We modify the experiment from Drurik, and then we use crown grafting, which is pretty much putting, this part was an infected road rosette from knockout. And then you cap it with one of the 1,000 pipettes. And then you can see that the plants that were inoculated barely developed the roots compared with the healthy. And we saw the callus initiation, but doesn't happen. So pretty much what you guys said yesterday, that the virus is actually moving down. Okay. So the question was how we can deal with the asymptomatics in the field. And that was a question that in Europe, the people is quite concerned, how we can check an asymptomatic plant, because we don't want that stuff here. And in our fields is, we have a lot of asymptomatic plants in our experiments. And so how we are going to sample them, because we know that it's not uniformly distributed. So we decided to look at the plant from the top. And these are rose plants seen from the top, and then we divided in 4 quadrants. And in the very first experiment, we start collecting 2 samples, and but we target initially only infected plants, and we wanted them asymptomatic. I'm going to show you some of that. So after that, we had all these PCRs that vary from 8 to 12, depending on the experiment that we were doing. So in the first experiment, you have here 3 plants, and all of them were with symptoms. But of course, the symptoms are not in every branch. And then you're having here, for example, quadrant 1, and then you have the asymptomatic, the symptomatic, asymptomatic, symptomatic, symptomatic. So we were collecting that. In this plant, you have 4 that were asymptomatics and 4 that were asymptomatic and symptomatics. So at the very end, and now we are in the statistics process and designing more experiments and repeating these things again and again. And then it turned out that we say, okay, this is with positive plants. Let's go now with the asymptomatic plants. And we came out with another experiment in which you have the plants that were inoculated. Someone is having open the microphone. Thank you. So you're having here the plants that, again, the quadrants, the plants, all of them asymptomatic, but they were inoculated by grafting. That happened in November, and this was January, February in the glasshouse. Okay. Then and then well, all these city values, PCR stuff, average, and then you have, you know, these plants, you have some positives and much negative, but you got a positive. In this plant, you got again some positives, positive and negatives, but all of them are symptomatic. So we wait until the plant was symptomatic, and we went back to the same plants, and we went up symptomatic now. And we did the same sampling, and then now you got all of them positive. So now we're working the statistics with this, and thinking with Nar and Francesca that we are going to move now out of my field. And then, well, the thing is that you can collect between, or you can test 8, 12. This is the number we are playing with statistics, and perhaps less than that. But if you do a survey that type, apparently we are in the way to say, okay, this plant is infected, but it's asymptomatic based on statistics. So this is something that is coming. We are not done yet, but just wanted to share you some preliminary results. So let me talk now about EDNA. And but before that, you need to understand EDNA. And EDNA is not what you think. EDNA is actually a method that started 17 years ago. Seems yesterday to me, but I'm done with it. But it's addictive. So we are still working with that. So EDNA is electronic probe. You listen to me talking about e-probes, diagnostic nucleic acid analysis. The original idea was a concept that came out of Bill Snyder. At the time, he was working in the USDA, and he teamed with us at the Institute. And we noticed that the idea was excellent. We wrote the proposal, got rejected, of course, because people was not prepared for this. And then good critic anyway, so we worked it out, and then we got it again. From the original team 17 years ago, there are only 2 guys working in that. The others retired, some other people move out, some people resign. So at the very end, I say, I'm going to continue with this because I used to finish better stuff. So at certain point, too many software, too many languages, bioinformatics. I'm not a bioinformatics, but I work very well with bioinformatics. I understand them, and I know that they speak a different language. And then Andres and I have been working together, and I told him, this is going nowhere. You know, if you don't convert this in Windows, let's close the door. So it took him 2 years to make the Windows thing. And he did it. And then we got a new director, and she has a vision of business. And this is what is called now My5 Tech. So if you go to the webpage, you register, but be careful because My5 Tech is by pay because you're supposed to use large volumes of data. And usually companies and some projects do that. But if you are a researcher, poor like me, so you can go for the free one, which is the Wi-Fi Legacy, which is the old server. It's slower, but good. So pretty much, EDNA is a combination of software. Is my probe that allows you to design the e-probes, my detect that detect with the e-probes. And now we publish MISTANK, that is going to be the next addition to, which is a simulator of HTF. So what is EDNA? EDNA, this is the in silico part. This is what you're supposed to do when you're developing an EDNA. So you create database. Pretty much a database of virus genomes, in this case, host genome, and the virus taxon we call new neighbors. So the taxonomically related viruses to the targets, or other diseases that may be infecting there too. That because when you do HTS, it will be practically nesting everything. Then you go to my probe with these databases and generate the e-probes, and they need to be of different sizes: 20, 40, 60, 80, and then you start trying. Once you have all these databases, then you have to curate them because many of them are not specific. And then you get your curated e-probes for virus, host, and then e-neighbors. And it's when you are ready for simulating. So you go to the host genome and you spike it with the e-probes that are curated already from viruses. And then you go with the HTS simulation. In the very beginning, we used medicine. Now we are using our own because it's more adjusted to and more precise. And then allow us to design serial dilution experiment in silico. This segment, depending on how dedicated is the operator, may take from 3 to 6 months to make a EDNA in this way, until here. And the last we did was for 46 viruses in soybean, and it's published. It's working a lot. The problem is validation. Having all these 46 viruses have been difficult, so we are at the point of we validating with 9. So, but it's coming along. It's beautiful. And now we're working with cancer. We have been trying with two genes of cancer, breast cancer, BRCA1 and BRCA2. And now we jump into TP53, which is the marker that we're using. And this is another problem, but it's applicable to what you're doing in QTLs because are single mutations. Then we don't have the software, my probe, for that type of problem, but we are understanding what to do. So after that, you are ready for in silico because all these curated e-probes go and are loaded into MyDetect, which is the other software of the modifier. So now you are in vitro, and then you go regular, nucleic acid extraction, library preparation, all this, and you do the HTS, and you have 2 options at the moment. Mini ion, Oxford Nanopore, or Illumina, and then you get your metagenome, which is the one you load in MyDetect to be screened with 8 probes, and then you guys just know. And it's working very nicely. It's a very good diagnostic method, and it's expensive, of course, because this is expensive. But at the moment, the prices are going down and working with mini ion. Perhaps this is applicable at the foundation blocks or wherever is your material is of value. And okay, so this is EDNA, and it's nice. In the case of QTLs, and because the statistics of EDNA, what we are doing is taking the mutation of interest and moving it in the e-probe in different positions. So the software recognizes them like a different thing. And you have to have at least 20 of this to have a good detection. But it's very time consuming, it's annoying. And our student has been complaining. And I've been buying a lot of good coffee from Colombia. And having parties at home and then forgetting about it. Now let's talk about another girl. So this type of things. Okay, now this is the validation of limit of detection of EDNA. This was published this year, and this work was done by Lisbeth. Lisbeth worked with us already, but she's now a professor in Ecuador. So, and then pretty much what we did was we created this artificial positive control, and these blue lines are the places where the e-probes are binding. So we did now serial dilutions of a plasmid, and then repetitions, statistics, all of this is included 3 times. This was a very expensive experiment and lots of full of hair when it was built for this. And now we know that different limits of detection has to do pretty much with the e-value of the e-probes. And then you define different levels, and pretty much they are in between picomoles, nanograms, and you can detect 10 copies of a virus. So it's pretty much similar with PCR. But again, variability between one user and the other operator. So we are still far away in the way that we need to do these circle experiments for future validation and these type of things. But the technology is working nicely in the lab. Okay. Let's go to the field now. So we are at OSU Mingo Valley. This is near Tulsa. And we picked this lot because it was very uniform in the soil. And we planted in 2022, the first 800. The plants are coming lovely. It's a show in the springtime with all these flowers. I'm not happy with my the way OSU is supporting me because I need a better field, according to me. I saw the pictures that you guys were showing yesterday that are real gardens. And I want to get there. So the public can go and visit. And this is Carmen. Carmen is the doctoral student, and she's looking at one of the first of the three that we got this year infected with rotoscopy. And of course, this I have to acknowledge Akhtar because Akhtar has been very supportive. Every time it's raining, you know, I don't want even to think about it. And then usually Akhtar arrives first. And so what we're going to do with all of this? The idea is that initially we can apply EDNA and HTS at the breeding blocks, perhaps foundation blocks. I don't know if you heard about this unit, the Bento PCR moving equipment. And this is very nice. It allows you to do extractions, PCR, gel, everything you can do in here. And it's quite portable. And so we want to use this because with this one, you can do actually the library preparation in the field and then run in the HTS. And we are doing PCRs, we are doing RPA, all the methods can be done in here. And this is allowing us to gain a lot of time and decentralize in PCR. So now we're moving to the rapid methods like this, in which you can see 2-3 lines, and then you say, okay, possible. Then this is working nicely. We are conjugating RPA products, PCR products, LAM products with lateral flow devices like this. And you can get this done in 10 minutes. And you can have the reaction done in here in 1 hour. So pretty much in the field, you can get results like this in 2 hours. From sampling and collecting straightforward in the PCR tube, like in the very beginning. And this is it. So I'm ready for questions.
All right, thank you. Do have, I guess, some time for questions. If you have them, go ahead, please.
What is the cost of the lateral flow?
This one.
Yeah.
The number so far is that this is about $10 to $12 per sample. But I think they can be adjusted. It depends on the volume of PCR tools that you buy. This is the one that is with the cassette, which is the most expensive because my lab is following the One Health. So we work with animals and humans as well. So we develop diagnostic for the whole thing. So the idea is working with plants in the very beginning, saving a lot of time and money, and then jumping to animals and humans. So but this is the most expensive. But there is one that costs probably half of this. Then it's just a chip, like the idea.
Right. I was just wondering how that compares to, let's say, standard immunoassay for virus diagnostics.
Well, everyone is having a different price. My colleague in the animal diagnostic lab, he told me every time we write a proposal, is $50 per PCR. Jen Olson told me $35, but of course she's putting overheads and all possible prices. That is the right thing to do. I don't have these numbers. I have just, I would say, basic numbers. And because at the very end, salaries and overhead change
So,
place to place.
so real quick, I think you can answer that. Your estimate of the cost includes what?
No, no overheads.
So just reagents
Just
and
reagents.
consumables, correct?
Yeah.
Okay.
Yeah.
For how many samples?
Well, usually I would say 100.
Okay.
Then, and then this is about 10 to 12. I think we can drop it to 8, but doing some changes. And then PCR for us is about $5. And lamp is a bit expensive too. It's about similar to this. But I don't like lamp. I just prefer RPA. RPA I think is much better. And and now we're making numbers for CRISPR-Cas, which is also a bit expensive, but it's coming along. So yeah.
The reason why I asked this question, I was wondering, in the application of it, if, a breeder or a company wants to test their plants along the line, how much would they be willing to pay for testing like this? So I was trying to get an idea of.
Yeah, that's why we're thinking in that unit, because that unit at the moment is $2,000. And you have everything in it. So I think if we're transferring technology to the farm, it's not only about telling them this is the way you have to do it. The hands-on training. It's about the equipment that they can use too, preparing a budget and saying these are the numbers, and decentralizing the diagnostics labs, because they may take their own decisions in the field without waiting for a week. And that's a big thing if you have an epidemic or something. So this is pretty much the idea. So at the very end, depending on the size of the nursery, they will decide, no, I'm going to pay for this, or I'm going to do it, or me, my neighbor, and the other guy in the other county, we are going to do it together. So these type of things. I think at the very end, we need to present these technologies to growers associations, stakeholders in general, and then say, well, you know better how you to handle this, your needs, and then we're talking about tailoring this type of technical transfer.